And so in this chapter, we're going to be thinking about the arrangement of branches, the shape of woody tissue. And for me, that really means going to a soft spot of mine, which is thinking about trees. And I think that in many ways, trees are such an excellent reminder and conceptualization of time, and that they experience the landscape and yet, unlike an organism like a small animal or a bird, they're not able to move if conditions are maybe too hot or too cold, or the habitat in which they first started to grow has changed. So what we see in the form and shape of a plant like a long-lived tree like this bristlecone pine is really the story of its entire life. And I think in particular, something that's always fascinated me, particularly with those trees that grow in California, is the incredible diversity of what the bark will actually look like on our trees. I feel like in a lot of ways, bark is like looking at somebody's face and their skin, and that it's very much the same way in which a tree wears that tissue. So if you look at someone's face, you might see lines of worry above the brow, or maybe places around the eyes because there's someone who smiles a lot and likes to laugh. Or maybe you're seeing the signs of sun damage or smoking. So when you look at someone's face, you see their life story, you get a sense of what they've been through. And the same is very much true when we look at the bark of a tree. There's a story that's told in that woody tissue. Looking at Abies concolor, which is white fir, and this is common for a lot of trees that are conifers, you can notice actually vigor and growth just by looking at the way in which the bark is exposing those redder grooves. So as that bark ages and expands, it oxidizes and turns that grayish color. So looking at that tree on the right, or excuse me, on the left, we're seeing younger bark tissue that's being formed and emerging. And it's telling us that tree is expanding maybe quicker than that tree on the right. Something else that's always fascinated me is alder. Red alder is one of my favorite deciduous trees. And I noticed that when you look at the tissue, bark tissue, you find an incredible array of epiphytes that grow on these trees. And those epiphytes are speaking to different local climatic conditions. And we can see just from these epiphytes maybe how humid the environment is, how much maybe airborne pollution exists in that habitat. Some of the trees that grow in the more drier areas of the range may not have epiphytes at all. They have this grayer bark. Some of them, though, have this incredible orange color or appear bleached white. So all of that are things that really speak to and tell us about the environment just by looking at the texture and surface of the tree itself and what is able to grow and live on that tree surface. Some other examples that have always fascinated me when looking at the structure of the tissue of a plant. Here we can see a redwood tree with a hollowed out scar in it, what we call sometimes a cat's face, though in these larger examples, they're often called goose pins because early settlers during the gold rush would keep geese and other livestock in these little corrals inside of trees. And that really tells you about that history as well of disturbance and fire that organism's experienced. You can also see the effects of humans as well. So here's an example of carvings back in the 30s made on a Populus tremuloides aspen by someone who was a little bit bored, I guess. I heard that these are left by Basque farmers in the Kings Canyon valleys. And you can also see the effects of pestilence as well. So we can see these pitch tubes on a pine tree that speak to mountain pine beetles burrowing into the tree and then being forced back out by pitch and other pus equivalents that trees produce to drive organisms that invade them back out of the tree tissue. And we can also look to the branching patterns as well. And you can see on the left, this tree that has this incredible spiraling to its stem. And that speaks to how there's been phototropism. So an effort to put branches on a side where there's more light exposure. And then this tree probably was growing in a valley where there was a lot of wind coming in a particular direction. So having wind then pushing on those branches and essentially twisting the shape of the tree so that branches continue to be applied on the sunny side and the wind continues to kind of push those branches sort of like a paddle wheel. You get this incredible twisting form in the woody tissue. Not all trees will be growing in this kind of erect pattern like a conifer. You might also see something like Aesculus, where the branches are very diversified in the direction in which they point. And that brings us to this idea that when we think about growth patterns in plants and anything that's essentially growing taller than just grass or moss. We're thinking about plants that are doing this in the service of, again, those leaves and that tissue that will be responsible for photosynthesis. So I want us to go back to what we discussed in our preceding chapter and think a little bit more about leaf tissue and what that tells us about the architecture of our plants. So here are three conifer species that we find in the Sierra Nevada, the Western Sierra Nevada, and Ponderosa pine and Abies magnifica are both in the Pinaceae family. So that's the pine tree family. They have these well-defined needles. Chalcedrus decurrens, incense cedar, has more of this owl-shaped kind of rectangular structures that it builds. And all of them have designed their structures to affect the needs that they have of accessing light. Interestingly enough, if you look at these tissues, you can find the years of growth of those needles and how long they actually hold on to that needle tissue. So if we look at our Abies magnifica, every time there's a branching point in that growth, we can roughly say that that's about a year's worth of tissue that was added to that tree and a year's worth of needles. So we might look at this and say that we can count back maybe 10 or so years just from this picture. So we can actually see needle retention and years in the physical structures of the plants. Looking at ponderosa pine, notice how those needles are all out towards the end. So if you were to zoom in on that branch, you would see bud scars, areas where the needles had fallen away, and it would show you that those needles only reflect about 1 to 4 years of growth. Chalcedrus decurrens is a little bit harder to use this principle, but in terms of needle retention, it's about 6 to 10 years. And so this tells us two things. It tells us first that there is greater shade tolerance among things like Abies magnifica than ponderosa pine, and that's because you can imagine that if you're growing a leaf tissue and you're building it to last over a certain period of time, you need to build a tissue that will be able to tolerate extremes of sunlight, but also the minimums experienced when there's shade. And so a fir tree like Abies magnifica grows in the shady understory. It's always adapted to that shade. And we can imagine that those needles that were applied maybe 5 or 6 years ago, as more needles are grown, those older needles start to be in the shade of the rest of that branch and of other growing branches. So if we're going to still have that needle be something that can effectively provide us with photosynthetates and sequester carbon for use in sugars and growth, then we need to build a needle that will be effective at shady environments. Ponderosa pine, it loves the sun, it grows in the sunlight. And so as a result, when we look at a tree like a ponderosa pine, we find that after only a couple of years, those needles are dropped because they were not designed to exist in the shady areas that are created as the tree continues to grow. And then Chalcedrus decurrens, again, is kind of that middle ground between shade tolerance and sun tolerance. And it is also a different family. So we see some other attributes that exist with Chalcedrus decurrens. But this is also interesting in thinking about another aspect of structure to our tree and architecture and branch pattern. And that is, especially in our Sierra Nevada ecosystems, a tolerance to wildfire. So we can actually see that when we have needles that are held longer by the tree, that also means we have branches that are held longer, because those branches, if they still have needles that are live, they're not going to be dropping those branches. And so with Abies magnifica, as opposed to ponderosa pine, we see that in a mature tree, we have a very different amount of space between the understory and where the live crown starts. And we also find that for things like ponderosa pine, and also to some degree Chalcedrus decurrens, we see that there is drop of dead branches, so a self-pruning that takes place. So recall earlier we talked about in our section on fire ecology that some plants, in particular plants that are adapted to fire, may have fewer what we call ladder fuels, fewer low dead branches that can convey fire from the understory up into the canopy. And that's what we're seeing with things like ponderosa pine and Chalcedrus decurrens. Whereas Abies magnifica, though also adapted to these spaces, has maximized growth in shady conditions and is therefore limited for its ability to grow in spaces where there will be repetition of fire and those fire cycles that could essentially kill this tree. Again, because I mentioned that Chalcedrus decurrens is in a different genus, it is also lumped with redwood trees. And what we find is they have really thick bark, just like a redwood, that can also help to dampen effects of fire damage. So worth pointing that out as well. But something that we'll see here then is there's kind of this contrast between maximizing for shade versus maximizing for fire. And so what we call this then is an ecological trade-off. And so what we see is that where there are traits that are beneficial, and yet in developing that trait, those two traits come into conflict, we have certain restrictions. So you can see on this axis that as we develop trait B, we sort of lose out on how well we can develop trait A. And of course, we could, in theory, maybe have a different way to develop both trait A and both trait B, but it's possible that may simply be a limitation in terms of what the plant can do without having some sort of limitation in its capacity to function in that system. Or it's possible that if we don't maximize one or the other of these traits, that this species will no longer be fit for its environment. So what we're seeing then is that there is, in this case, but frankly, in many ecosystem interactions, we find that species will have trade-offs where they'll have to balance things like fire and needle retention, or often we see a balance between shade tolerance and drought tolerance. So things to pay attention to and identify in the systems that you work are those sorts of trade-offs between different attributes that may at times be in conflict. We've thought a lot about the outward manifestation of age and time and environment on our plants in terms of bark and branch structure and now how that reflects leaves. I also want to kind of turn things a little bit inward. So I'd like us to think about what that woody tissue itself is actually doing in our plants. And of course, in many ways, it's a supply line to connect our leaves with water. So recall that leaves are not just utilizing CO2 from the atmosphere, they also need water, which they're getting from their roots, and coupling with that CO2 to form glucose. And that glucose will be what we use to power our cells, but also to build things like cellulose, which is essentially the woody tissue that our plant is constructed of, and that we also use for the construction of the carbon aspects of our leaves. So we have this stuff called xylem. It is the conduits that move water and also minerals from the soil up to our live tissue. And then we also have phloem, which is moving sugars from leaves to other parts of the plant where construction is required, such as moving them maybe to the roots or moving them to areas of new growth or to help reinforce living cells. So let's think about what that woody tissue looks like in a cross section. So if we were to take a cross section and say we're cutting it perpendicular to the growth of our plant stem, and we looked at it during those first nascent stages of what we call primary growth, we would see that the tissue is mostly wasted space, you might say, with this area known as vascular bundles, which include that phloem, so again, moving sugar, xylem, moving water, which are sort of floating around and distributed in a loose ring within that tissue. So you might think then that there's a lot of this wasted space here, but essentially that area out to the edge, the cork cambium plus paradermal, epidermal tissue that grows there, and also this parenchymal stuff and the pith, that's serving as packaging. So imagine if you were going to send a Ming Dynasty vase cross country, you could probably just throw a postage stamp on it and write your name on the vase and put it in the mailbox, but it's going to get broken when it shows up where at the address that you've sent it to. So what you do instead is you get a box and you put a bunch of packing popcorn in that box, and that keeps your vase safe. And so what the plant has done is in those early stages of growth, it wants to protect the very vulnerable vascular bundle. And so it's using the parenchymal and the pith and also material known as cortex to essentially be packing popcorn, and then it's putting it in this box, which is that tissue grown from what's called the cork cambium. So we have this thing called a procambium, and you'll notice that it appears in picture 2 as well. That procambium is meristematic tissue from which phloem and new xylem tissue will be grown. And we continue to add more and more new xylem and new phloem until these vascular bundles start to bump into each other. And it's at that point where we have run out of space within that ring shape to add new vascular tissue, a new phloem and xylem. And so at this point, we have essentially our first growth rings. And from there, we can start to add what we call secondary xylem and secondary phloem, which would be that initiation of a second year of growth of these tissues. And what we'll find is that the direction of growth is always coming from what we call that vascular cambium, so that ring of meristematic tissue producing phloem to the outside, xylem to the inside. And so throughout the year, we're adding more xylem. And xylem, again, I said, are these conduits for moving water throughout the body of the plant. So you can imagine that, if you live someplace that's very busy. You might have a 4-lane highway to move lots of cars. If you live on a place in an area that's rural or where there's less need for commute, maybe narrower roads. So at times of the year when we need to have more transport of water, the xylem cells that we're creating are going to be larger in diameter. And then later in the year, at times where there's less growth, those xylem diameters, those conduits are narrower. And so what we start to see, as you can see in this pattern here, is this variation of bigger diameter xylem and smaller diameter xylem. And you'll see this also repeated in a phloem, but we're just looking at xylem right now. And that pattern is what we see when we're not looking under the scope as patterns of growth rings. So looking at that secondary growth, now we've had years of growth occurring on our tissue. Again, we'll still see that there is this vascular cambium that separates growing new xylem tissue. And it's worth noting that xylem tissue isn't functional until it's dead. So we have to have a tissue that's grown and then hollowed out to allow for water to move through that hollow tissue. And then to the outside, we have a phloem, which also is being continually grown from the vascular cambium. And because that tissue, as the vascular cambium adds more xylem and phloem, continues to expand, what's happening is our wood, our xylem, continues to grow in diameter, but so too does our phloem. So the oldest phloem, which is on the outside of this cross section here, will start to break and shred apart. And so when you look at a tree and its bark and you see that shredding, breaking pattern of the bark, what you're seeing is the oldest phloem on the outside that's being ripped apart, the younger phloem towards the middle, which is about smaller diameter, so less of that shredding occurring. And then with phloem, unlike xylem, phloem needs to be live in order for it to be active. So we have to continue to add more phloem and have companion cells associated with that phloem to help with the movement of our sugars throughout the tissue. So we then see these growth patterns within our wood. And this is kind of a zoomed out view of that earlier photo that we saw looking at the xylem up close, where we see those kind of, I guess, lighter areas, those kind of peaks in this cross section of wood. That's where there's a higher concentration of cellulose. In other words, a higher concentration of cell walls. And again, that cell walls and high concentration means that that's where we have a aggregation of those smaller diameter xylem cells. And then those areas that kind of, excuse me, dip within the areas between those peaks, that's where we have congregation of our cells that are of larger diameter, so less cell wall tissue there. So we can take this and then make extrapolations through a process called dendrochronology to infer our understanding of the past. So for example, we might take in this cartoon a cross section of a piece of wood that measures 19 rings, and each of those rings would speak to a year of growth. And we might then think, well, when was this tree alive? When was it growing? So notice that there's variation in the width between those patterns. So if we had a year that was very wet, we might expect to see more growth. And so more separation between those rings. If we have years that are drier or there is some pestilence, maybe we see less growth in that year. So maybe the rings are closer together. So we can see variation here, and we can start to match these up almost like barcodes. So I've got this other piece of wood that we found, and we know that this one was cut down in 2020. So we can say that we have an idea of when this was felled, but we don't still know how that matches up with that tree that's 19 years old. But notice that we have overlap here between these 3 really tight years of growth with 2 longer periods of growth on either side of that. So that might tell us that, and if we know that these trees grew in the same habitat or location, we might be able to say, okay, these trees were experiencing the same sorts of climatic variability. We might be able to say then that we can match these up and use these trees, using our tree that was cut in 2020, to infer when that 19-year-old piece was cut down. So let's do that. So here we can count then back 11 years and say, all right, so we know that this other tree, that piece of wood speaks to a period of time in 2009. And from that, we can then get a sense of when those rings represent in time that they were growing. So this is a approach in dendrochronology that is very useful for folks who work in archaeology. It's been used a lot in the Southwest to help to identify the age of pueblos. It's also been used in Northern California to identify the age of some of the plank houses built by Yurok and Karuk tribes peoples. And it also has a lot of applications as well in the art world. So folks who are trying to figure out the provenance of maybe a painting, they'll look at the wood that's in the stretch bars of a canvas and use that to kind of figure out if the piece is a forgery or not based on tells of the wood tissue. If you work in carpentry, you probably also know that not all trees will have growth rings that look like the rings that I've shown you. So these are rings from conifer trees. Different plants will have different features. In particular, trees that we consider to be hardwoods have different xylem structures that are present with as well within that tissue. And that means that it can be possible to identify trees not just with respect to age, but also to species. So things like oak, for example, you can see these burrs that exist within that tissue, which are structures called arrays that help to move liquids not only in a vertical fashion through the tissue of the wood, but also horizontally through that wood. And that's something you don't usually see in a conifer. But I also want to mention, in thinking about this, that all of this tissue can also be used to think about climate data as well, not simply in the more coarse approach that we're taking, but you can actually look at the carbon and also oxygen molecules that are trapped within this woody tissue and use that to extrapolate trends in climate, such as temperature of a given day or how much precipitation was received. And that gets into a territory that I won't be covering in this class, but I've included links about, which is stable isotope ecology. And that's been a really important way that folks have utilized wood in places where they may not have access to things like ice cores to make sense of past climatic events. I'm always amazed by the texture and quality of different types of wood. There's a building I work near on campus called Mulford Hall. And if you get a chance to walk into Mulford Hall at UC Berkeley, they've got these incredible displays of cross sections of trees from all across North America. And it's a great opportunity to see the diversity of wood and function and form that exists in North American forests. I think that that tissue also speaks to our role in our history in all of this. So I think I just want to end by mentioning in this section that when we look at a cross section of a piece of wood, there is so much of a story that is told by that. One of the older cross sections, and that is for me the most astounding that exists in Mulford Hall on campus, is a redwood tree. And that redwood tree measures about 7 feet across. And if you count back those growth rings, you can go back almost 1,000 years. So it's a pretty old piece of wood. It was felled in 1957 up in Humboldt County, where I'm from. These are pictures on the right of a historic photo of trees, redwoods that were cut down in Northern California near where that tree was felled in the 1800s. So you can see just their incredible mass and size. You don't have to count back though 1,000 years to get a sense of our role in our history and all that. For me, it's just a knuckles worth is my lifespan. And I say that just because in just a quarter century, we have affected incredible change on our forests. We've almost doubled CO2 emissions into the atmosphere. Globally, forests have been cut into a quarter. So these tree rings, they don't always represent the most clean image of the past, but they are honest in describing the effects of the past in the present and also our effects as humans in these spaces. And what the rings of these forests will represent in the future is really something that we get to decide as stewards of this landscape. And with that, I have a couple images just of forestry tools that are commonly used for assessing age and taking measurements. Because I know we're getting a little pressed for time, I'm going to go to questions, but all of these tools are outlined in the follow-up resources that I've included. And while these tools are sometimes expensive, I know that they also are tools that are sometimes more available to folks who work in restoration. So that's why I've included these as options for you to explore if this is something that interests you. And with that, I will turn things over to Amy.